Okay, so tissue fluid. Tissue fluid is almost identical in composition to blood plasma, except there are fewer protein molecules and no blood cells. There are some white blood cells because they migrated in from the blood on purpose. But how much protein and how much plasma or how much tissue fluid there is will depend on two opposing pressures. So how much pressure, how much tissue fluid you have, the more blood pressure at the arterial end of the capillary you have, the more tissue fluid you will have. The more osmosis you will have drawing tissue fluid away, the less tissue fluid you will have. So osmosis will draw tissue fluid out, back into the venule end and through into the lymphatics. All right? So we have the tissue fluid, the arterial pressure forcing tissue fluid, forcing plasma out of the capillary into the tissue fluid space. And we have osmosis drawing tissue fluid into the lymphatic vessel and back into the venule end. So those are the two opposed forces, blood pressure and osmosis. Those are the two opposing forces that are balancing tissue fluid. That's the scales being balanced here. So the exchange of materials between cells and the blood is what tissue fluid is for. It's important for the environment that the cells need to work in. It's important for homeostasis. Homeostasis is for the maintenance of a constant internal environment. The regulation of the glucose concentration, water, pH, metabolic wastes, and the temperature all need to take place to maintain the composition of tissue fluid at a constant level. And each individual tissue bed has its own balance of these factors to maintain. If these factors are not maintained in balance at each tissue bed, and each tissue bed having its own certain balance of factors, then you will have a rough day, to put it in rough terms. Now, let's talk about lymph. I mentioned lymph vessels a couple of times now. 10% of your tissue fluid is collected and returned to your circulatory system through lymph vessels and lymphatics. A lymphatic is a tiny blind-ended vessel with valves that are wide enough to allow for a large protein molecule to pass through, but not so wide as to allow, for a large cell to pass through. So little protein chunks can get through, but cells can't. So yes, no. All right? Now, lymphatics are found in nearly all tissues. Anywhere you have capillary beds, you're probably going to have lymph. So edema, which the British spell O-E-D-E-M-A and Americans spell E-D-E-M-A, is going to be the buildup of tissue fluid due to an imbalance of protein and a rate of loss from the plasma with concentration and rate of loss from tissue fluid. So whenever you have too high of a blood pressure and not enough osmosis drawing that tissue fluid away, or you have a blockage in the lymphatic ducts that's allowing too much fluid to accumulate, or any imbalance that causes fluid to accumulate in the tissue beds, that's going to be edema. And there's different kinds of edema, but in general, it's just an accumulation of fluid in the tissues. So lymph is the fluid inside the lymphatics, and it is identical to tissue fluid because it is tissue fluid. It just came from tissue fluid, so it's tissue fluid. And lymph nodes are those nodes along your lymphatic tracts that, along your lymphatic vessels, that are involved in protection against disease. You have these lumps of tissue along there that will have a lot of immune cells within them that are tasting this tissue fluid and deciding whether or not to respond in an immune way. So these lymph nodes are found at various stages. So edema, it's a bit tough to see in these particular images, but safe search on, you have access to a number, well, no. Safe search on, you have access to the internet. Type in edema. I'll wait. Now, so this is a better image of how our arterial and our metarterial and our thoroughfare channel and our capillary bed and our tissue space and our lymphatic ducts interplay with one another. So this is a more complete diagram. Now, this is a diagram that shows the lymphatic system in basically a Ken doll. So the largest lymphatic vessel is the thoracic duct. And that's found in the chest. It runs adjacent to the inferior vena cava. Maybe not adjacent to it, but nearby. It's nearby. Anyway. Now, red blood cells. Let's talk about those for a moment again. The pigment hemoglobin that I mentioned before, it is a globular protein. Its job is to transport oxygen from the lungs to the respiring tissues. The stem cells that form hemoglobin, along with the stem cells that form every other white blood cell, are found in the bone marrow. Bone marrow is found in a lot of different places, and not even sometimes the bone. Sometimes it's also found in the liver. So it's also found in the humerus, the femur, the skull, the ribs, the pelvis, and the vertebrae. So eventually, red blood cells, which don't live very long, they'll live maybe 4 months total, including 1 month as a reticulocyte that I mentioned, and 3 months as a mature red blood cell. And then they'll become kind of stiff and they'll become kind of stiff and they won't bend anymore until they reach a tight spot in the circulatory system. And they'll find that tight spot and then they'll rupture. Usually that tight spot is the spleen. The spleen, which is right around here. Now, this is the bony trabeculae. These bony trabeculae, this is a space in the bone where the bone marrow is held. Now, red blood cells, they have an unusual structure in that they are very small. Their diameter is around 7 micrometers. I mentioned this before. They have a biconcave disc shape. They have no nucleus, no mitochondria, no endoplasmic reticulum. All they have is hemoglobin, maybe a little bit of a cytoskeleton. All right. Now, white blood cells are also made in the bone marrow from the same hematopoietic stem cells. They are very different from red blood cells in that they do have a nucleus. They are mostly larger. Lymphocytes are around the same size as red blood cells. But white blood cells tend to either be spherical in shape or irregular. Now, we can break down lymphocytes in that hematopoietic sense by granulocytes, agranulocytes, as I showed you, or we can break them down by their function, by phagocytes or by lymphocytes. Now, phagocytes, which can be classified, the neutrophils are also phagocytes, and monocytes, which can differentiate into macrophages and dendritic cells, are also phagocytes. And then lymphocytes, which are, well, lymphocytes. Phagocytes will tend to destroy invading organisms by phagocytosis, which we discussed in Unit 2. Lymphocytes will destroy microorganisms by secreting chemicals called antibodies, which will attach to and destroy invading cells. They'll be, well, lymphocytes. Now, this here This is probably an eosinophil because it is small. It is relatively small. It is a bilobed nucleus, and it has eosinophilic staining granules. Now, 50 to 70% of all of your white blood cells are neutrophils. Their main method of killing invading pathogens is by phagocytosis. Mainly they deal with bacteria and cellular debris. They are very important in the process of inflammation. If you've ever had a really satisfying pimple that you've popped, most of that pimple whitehead, most of that pus, is neutrophil carcasses. Yep. Eosinophils are 1 to 4% of your white blood cells. They help break down blood clots, and they have a role in killing parasites. Basophils are less than 1% of your white blood cells, and they synthesize and store histamine, a substance that is released during inflammation, and they also make heparin, which is an anticoagulant compound. Their other functions of basophils remain somewhat unclear. Now, agranular cells, like lymphocytes and monocytes. Lymphocytes are 25 to 40% of your cells. They function in the immune response, in the humoral immune response, through the production of antibodies. Monocytes are 2 to 8% of your immune cells, and they mostly do phagocytosis, typically as macrophages in tissues like the liver, spleen, lungs, and lymph nodes. So, I'm going to leave you after this spotter's guide by discussing thrombocytes again, and these are bits of broken up megakaryocytes. We've already really discussed these. Mostly when we bleed, the chemicals and substances called clotting proteins, prothrombin, will help to form an insoluble plug and seal off that bleeding point. Now, some of the related diseases of these blood cells are anemia, lymphatic filariasis, hemophilia, which is a bleeding disorder, AIDS, which is resulting from an HIV infection, which we will discuss in greater detail in Unit 5, leukemia, which we will also discuss in Unit 5, and thalassemia, which is a genetic disorder that results from a misfolding of the alpha globin. Also sometimes the beta globin, but that is less survivable than the alpha globin misfolding. And that's where we're going to end this video.